The overall goal of this procedure is to prepare recombinant adeno-associated virus 9, or rAAV9, to manipulate gene expression in mouse hearts.
The method can answer key questions in the field of cardiology and molecular genetics, such as how to study gene function in mouse heart in vivo. The main advantage of this technique is that it will allow us to easily manipulate gene expression and function in vivo in mouse heart, allowing the functional access of gene function without heavily relying on the genetically modified animal models.
After generating rAAV9 constructs and culturing HEC293 cells according to the text protocol, to transfect the cells on day 1, combine 70 micrograms of AAV rep-cap plasmid, 70 micrograms of rAAV9 plasmid, and 200 micrograms of ad-helper plasmid in a 50 milliliter centrifuge tube. Add 49 milliliters of room temperature DMEM without FBS to the tube and mix well. Then, to make a 4:1 PEI to DNA solution, add 1,360 microliters of PEI to the tube and mix well. Incubate the solution at room temperature for 15 to 30 minutes. After the incubation, add 5 milliliters of the solution to each 150 millimeter dish of